right, today's lesson has to do with the cell process known as meiosis, which is a type of cell division. It's a little bit different than mitosis, though, and we'll go through those differences here. But meiosis is the process of cell division that results in gamete formation. So you need to know what gametes are as a result of this. And there's a little bit of an overview on here on this front slide here. And this is an actual image, it's kind of cool, of a human egg and a human sperm. So you can see the difference between the two of them. So meiosis had to happen to get there, but fertilization is about to happen. So let's take a look at these processes. First, let's step back a bit. You've learned about mitosis. Mitosis is regular cell division. Normal body cells, how they make more body cells. And you learned all about PMAT, prophase, metaphase, anaphase, telophase earlier on in this unit, and then wrapping it up with cytokinesis. And so there's a couple of important, kind of phases of this that are relevant here. First of all, you had to have the DNA replication. Remember that happened back in interphase, not during mitosis itself. And then you had the whole phases of mitosis going on. This one right here looks a little bit like, I don't know, I guess it looks a little bit like metaphase, and this would be a little bit like anaphase. But in general, what you get as a result is two diploid cells. Now that's a new vocab word for you, diploid. Diploid means that a cell has pairs of chromosomes, one that originally came from mom and originally came from dad. But the key point is here, our original cell had two chromosomes, one red one and one blue one, and our final cells, this one and the one at the bottom, right here, both have one red one and blue one. So the key thing with mitosis, cell division, but yet on the same time cell multiplication, that you start off with a cell, you have to end up with the same number and kind of chromosomes when you're done. Mitosis does not work for gamete production. So if you don't know what gametes are eggs and sperm. If you're talking about reproduction, you can't use mitosis cells for that. So here's why. We've used these terms a little bit on the other slide as well, but we're going to use the letter N to represent the number of chromosome pairs, different kinds of chromosomes. Humans have a 2N number of 46. In other words, we have 23 pairs, but we have a total of 46 chromosomes, and our cells are diploid. In other words, each of the cells comes in pairs. So if our eggs and sperm were diploid, if they each had 46 chromosomes in them, after the process of fertilization, which is when eggs and sperm come together, that next generation would actually have 92 chromosomes, 46 and 46. And that would be something we would call tetraploid. It would actually have 4N, 4 of each kind of chromosome. But in a really short number of generations, the cells would be overwhelmed with chromosomes, because if we had 92 chromosomes in each cell, and in the next generation 2 of those came together, we'd have, 184 would come together. The generation of that, over 200 and something, all 300 and something chromosomes. So it just doesn't work for reproductive purposes. It works if a cell needs to make copies, but it doesn't work for sexual reproduction, where we're trying to bring cells from 2 different individuals together to make a new offspring. So for that kind of reproduction, a different process is necessary, and that process is called meiosis, M-E-I-O-S-I-S, meiosis. It sounds really similar to mitosis, so be careful how you say it. But meiosis results in the production of gametes. Gametes are eggs and sperm, eggs being the female gamete, sperm being the male gamete. And in those gametes only have one chromosome from each pair. When you only have one chromosome from each pair, we call that number haploid, like half, where each pair only has one representative. So in this case, fertilization works, because if we have a haploid egg that has 23 chromosomes and a haploid sperm that has 23 chromosomes, you end up with a zygote, which is a fertilized egg that is diploid again, and it has that original number of chromosomes. So it's also really important that when we talk about a cell that's haploid, we can't just pick 23 random chromosomes. It has to be one from each pair. So not just pick any 23, it's got to be a precise division of each pair of chromosomes into their partner, so to speak. So let's take a look at how this process works. Here's a general overview of it. Biggest difference that you'll notice in meiosis is there's actually 2 cycles of division. There's meiotic division 1 up here, and then there's meiotic division 2. They're really similar to mitosis in many ways. You still get PMAT. There's still a prophase, metaphase, anaphase, and telophase in each one, but it kind of happens twice. There's a first cell division and a second cell division. Overall important things that you need to know is that after that first division, you end up with cells that are haploid. So after the first round, so to speak, of division, we end up with cells that only have one from each chromosome. So if you take a look at this cell right here, this original cell started off with 2 chromosomes. By the end of it, each new cell only has 1 chromosome, and that's why they're haploid. In the second round of division, we divide up these chromosomes into their sister chromatids. So if you look at the beginning of round 2, there's only 1 chromosome, but each chromosome is kind of that X shape where it's 2 sister chromatids together. And by the end of that second round, we now have 4 individual cells with 1 chromatid each. That's the big thing that's different between meiosis and mitosis. So 2 rounds, an initial division that makes it haploid, and then a second division that separates the chromatids from each other. Let's look at it a little bit more in depth. Moving forward here. So here's the first round. I know it looks really complicated. Try to look at the big picture of it. So we're starting over here in the bottom left. Here's interphase, just like we saw before. This starts with a diploid cell. Again, it's got 4 chromosomes in this particular case, 1 or 2 pairs. So they've got 2 chromosomes from each pair. In interphase, the cells or the chromosomes DNA duplicates just like we'd expect. Prophase is the same as we'd expect. The chromosomes, they use the word spiralize here, but they condense into visible chromosomes. A little bit later in prophase is something unique, and that's why I put a big star by this. We get the homologous chromosomes, and they use the word synapse, but they come together with their partners. The partners are called tetrads, and the reason why they're called tetrads, if you look really closely, each tetrad consists of 4 chromatids, 1 from each or 2 from each pair, so that's where that word comes from. But that's the key difference right here, is that during prophase 1, tetrads form. The chromosomes find their partners. And then as a result, there's a big difference that happens here in metaphase 1, where the tetrads line up. In essence, they line up double file, not single file like they did metaphase. Here you can see there's a double file line. Here's a pair of chromosomes, and here's a pair of chromosomes. And that's the big, big, big difference. That's the really key thing, that if there's only one thing you take off of this slide, that's what you should write down, that at the end of metaphase or during metaphase, the chromosomes line up as tetrads. They line up with their partners. So that in anaphase right here, we can see there's the separation. Now the separation occurs not between chromatids, that's what happens in mitosis, but here between the pairs. The tetrads separate, so now you have 1 pair of chromosomes going north and another pair of chromosomes going south. So our end result up here is that we end up with, as the result of telophase and initial cytokinesis, that you get 2 chromosomes now in each cell. Each chromosome still doubled up, but each cell that's resulting here is haploid. So right here, these 2 new daughter cells, kind of at the halfway point, each one of these is haploid. They've got 2 chromosomes each, or half the number of chromosomes, but each of those chromosomes is duplicated. There's a lot that went on here, so you may want to take a, hit pause right now and write some of this down. I'm going to move on to the next phase. So this was the first cycle. Here's the second cycle. Cycle number 2 is essentially just like mitosis. So of the set that you've got going on, this one's actually a lot easier. So here are our 2 cells that came in from the end of that first cycle. And remember, each one of these is haploid, but each one of them has chromosomes that are still doubled up, consisting of 2 sister chromatids held together by the centromere. So what's going on next? Well, we still get the cycles of PMAT, prophase, metaphase, anaphase, telophase. Here you can see they kind of put a Roman numeral 2 after each one just to indicate that this is the second round of division, but that's no big deal. It's still prophase. You've got the chromosomes are kind of appearing. Metaphase, they line up. And now in this case, metaphase is just like what we saw in mitosis. They line up single file across the center of the cell. So this is really, really similar to what we saw in the last bit. Anaphase, the sister chromatids are now separated, the centromeres get pulled apart, and 1 chromatid goes north, 1 chromatid goes south. Again, just like in mitosis. In telophase, we get the new nuclei reforming, new cellular membranes, or nuclear membranes, and then we get that final round of cytokinesis. So what's the results? The results of this one is what you can see here. Now we have a total of 1, 2, 3, 4 cells. Each one of those cells is haploid, and each one of those cells' chromosomes is now made of just 1 strand, 1 chromatid. So we get 4 cells that result from this and 4 cells that are haploid. Again, a lot went on here, so you might need to hit pause and try to redraw this. But again, second cycle is pretty much just like mitosis. In metaphase, they line up single file. Anaphase and telophase progress just exactly like you'd think, but the result, again, a little bit different. Now we have 4 cells as a result, and those cells are all haploid. Overall, what are the differences? This is that compare and contrast slide. You know I'm going to ask you this on the test or on the questions when you're trying to demonstrate proficiency for this. So here, take a look on the left. We've got mitosis. You know the PMAT, all that kind of stuff is the same. In metaphase, the big thing is here that they line up single file. Here they are, right at one big row. They do not meet with their partners. They do not meet in homologous chromosomes. And then the results, you get 2 diploid cells. They should have the original number of chromosomes, and they should be identical to each other and to what the original parent was. So that's a key point of what mitosis is. That's how cells can stay the same generation after generation after generation. Meiosis is a bit different. Here's the key differences over here on the side, stuff that you really need to know. So first thing first, in prophase, we get the formation of tetrads. Again, the pairs of chromosomes come together, and you get now one tetrad over here, and then there's a second tetrad in this diagram on this side. The homologous chromosomes find one another and sort of overlap into a big jumble. Second big difference is in metaphase. And here you can see the difference here is that now they're not lining up single file like they did in metaphase over on this side. Now they're lining up with their partners as tetrads across the middle of the cell. And again, it's kind of a vertical line this way that they're crossing up. After we're done, you get 2 cycles of PMAT. Here's the end of the first cycle. Here's the end of the second cycle. And the results here are a bit different. We get 4 cells, not 2. And each one of those cells is unique, not identical. And those cells are haploid, not diploid. We would call these cells now gametes. Each one of these could develop into an egg or develop into a sperm. So overall similarities, the fact that the DNA replicates during interphase, the general phases of PMAT are the same. I mean, the things that occur during prophase or the things that occur during metaphase are identical. A little bit difference in how they line up, but that's about it. And then the cytokinesis is still the same. All right, there you have it. The big differences between mitosis and meiosis. Again, meiosis is gamete formation. If you're interested in earning a 4, here are some ideas of things that you can take a look at, kind of going above and beyond just learning the simple steps. And I hope you have some new understanding and recognition of how you came to be, I hope, and how we get some differences. As we go on, we'll take a look at some of those things and how this process of meiosis relates to these events of genetics that we see, like segregation, independent assortment, and recombination. See you in class tomorrow.